Hello students, this is Fadan Mirza. We are discussing the mitotic cell cycle today. To discuss mitotic cell cycle, we have to understand first cell division and what's the difference between a cell division and a nuclear division. For cell division, we need to keep in mind that cell division occurs because it allows growth, repair, asexual reproduction, and regeneration to occur. Cells are basically of two types, prokaryotes and eukaryotes. Prokaryotes include bacteria only, and you know that they lack nucleus. Eukaryotes, on the other hand, they have nucleus. They have a true nucleus. For example, protocots, fungi, plant, and animal. Protocots includes amoeba and plasmodium. We all have a true nucleus. The hereditary material in our cells is DNA and RNA, but primarily DNA. So the nucleic acid DNA is present in the nucleus in our cells, and bacteria is lying free the cytoplasm. Bacteria, how do they divide? They divide by binary fission. One bacteria will split into two, two will split into four, and so on and so forth. But eukaryotes, we cannot carry out cell division alone. We have to carry out cell division together with the nuclear division. True nucleus cells have to carry out cell division together with nuclear division. The nuclear division can occur by mitosis or meiosis. Mitosis occurs in body cell where the number of chromosomes remain the same. Meiosis occurs in gametes where the reduction division takes place and the number of chromosomes becomes half. This figure 5.3 is a simplified diagram of a chromosome taken from Mary Jones' fourth edition. You can see figure 5.3. These are two chromatids shown to you on a chromosome. They are attached at the centromere. Centromere holds no genes. There are no genes there, but it holds the chromatids together. So this is one chromatid. This is the other chromatid. Chromatid just basically mean how many DNA molecules are there. The number of chromatids is equal to the number of DNA molecules. If you are having two chromatids at the moment, it means there are two DNA molecules. And since there is a common centromere here, it means that the DNA are independent replication. So whatever DNA is present here, the same copy is present here. So the genes will be same as well. So this is a single chromosome. This is one part of a homologous pair. You receive this kind of a chromosome from your father, and you receive a homologue of this from your mother as well. So this is how homologous pairs are present. These are called as sister chromatids. Sister chromatids are always attached at the centromere. And sister chromatids form because of DNA independent replication. There are telomeres at the edge of the chromosome. They prevent the gene losses. This is the DNA molecule, black and yellow. I have shown this in two colors because the DNA is actually having one DNA molecule is having two strands, and this one strand will be, for example, black in color, the other is in yellow in color. So when it gets copied, there will be one black and one yellow, as I've shown in this key. This again taken from Mary Jones figure 5.4, fourth edition. You can see these are the nucleosomes, these are the histone proteins. They are actually eight histone proteins that form a nucleosome, and the DNA coil wraps twice around each to form chromosome. And this is the linker DNA, which basically is the continuity of from one nucleosome to the other. And this is how the DNA gets packaged into a chromosome. Chromatin is always the thread-like or network or granules because it's having DNA only. It condenses from chromosome. The chromosome will have one chromatid each because it will have one DNA molecule each. That it probably will not have the replicated DNA attached at that time. Why it appears chromatid and not as chromatin? Because the DNA is now associated with histone protein. So these are three different chromosomes shown to you, and each with a single chromatid. Now, when the DNA has been wrapped together with histone proteins, this DNA ends up being condensed together. That's why the appearance changes. If we discuss how a chromosome changes into a like chromatin changes the chromosomes, this is a chromatin, no chromosome. This was just granular structure. It still had 46 DNA molecules. It condenses to from chromosome with one chromatid. Again, 46 DNA molecules are there, but the appearance changes because of being wrapped around histone proteins. Then these chromosomes, the chromatid, one chromatid each, since there was a DNA replication which had occurred, so chromosome with sister chromatids formed, they joined its centromere. So instead of 46 chromosome and 46 DNA molecules on chromatin and 46 DNA molecules on 46 chromosome, you end up having 92 DNA molecules on 46 chromosome. Why 92 DNA molecules on 46 chromosome? Because each chromosome is not a single chromatid anymore. Each chromosome is two sister chromatids attached at the centromere. So the DNA, this DNA present here on this chromatid is present here, and its copy is present here. So the chromosome, when it gained this kind of an appearance where its sister chromatid is present, the DNA content of the cell becomes twice of. So if initially it was 46 DNA molecules, then once the DNA form, once the chromosome gets this appearance, you will have twice the appearance, twice the DNA content. So this when where this process occurred. For this, we'll be going to the details of the cell cycle. This is a mitotic cell cycle. These are the phases. The longest phase of cell cycle is interphase. You can see this on this pie diagram, again taken from Mary Jones. G1, S, and G2 are the phases of interphase. G1 is the longest phase, G2 is shorter, and S is the shortest phase of interphase. S phase is the phase where the DNA replication occurs, and M phase is followed by interphase. M phase is where the nuclear division by mitosis occurs. This is followed by cytokinesis, which is the cell division. As discussed previously, we are eukaryotes, so we have interphase, M phase, and cytokinesis, the cell division occurring. There are three stages in our cell cycle, but if it's a prokaryote, then the M phase would not be there. The interphase would be directly joined with the cell division, cytokinesis, and then again interphase, and this is how it will proceed. What occurs in each of the stage interface, basically, this is how the chromatin, the DNA, will be continuously in chromatin form in interface, whether it's G1, whether it's S phase, whether it's G2. Although in S phase DNA replication occurs, but the 92 DNA will still be in the form of chromatin. They will not arrange themselves in chromosome. They will not give the characteristic appearance of the sister chromatid joined at the centromere as yet, because right now it will all be chromatin, because the DNA, although after replication, it has not yet formed that kind of structure which is formed because of the arrangement around the histone proteins. So what happens in G1, S, and G2 phase? I've discussed already S phase. This is where the DNA replication occurs. In G1 phase, growth occurs, and RNA enzymes and proteins are made. In G2 phase, the DNA is checked for errors, and how the DNA is checked for errors? It is checked for errors using some genes, which are tumor suppressor genes. Tumor suppressor genes are already present in your cells. These include p53, these include BRCA1, BRCA2. So this basically works somewhere here, somewhere between S and G2 phase. So this is where the DNA replication occurs, and this is where the DNA starts to be checked for errors. Tubulin is synthesized in large quantity in this particular G2 phase only, because the microtubules will be required in large quantities in the cell once it enters into the M phase. Now coming to the M phase, the M phase is basically having four different phases, four different stages: prophase, metaphase, anaphase, and telophase. So how the cell changes during this entire phase, I've drawn some cells here. So in interphase, you can see a cell, and then you can see the nucleus of the cell as well. The circle within the circle represents the cell within with the nucleus. In S phase, again, there will be a circle, and within the circle, so that's again a cell with its nucleus. In G2, again, the cell will have its nucleus. In prophase, there will be a cell with its nucleus, but in metaphase, anaphase, the nucleus will not be there. And in telophase, again, in telophase, the nucleus will be not there, but it will start to reform. By the time cytokinesis occur, you will have two separate cells, each with a separate nucleus, and they again will enter interphase, and the process of DNA replication and the growth and G2 phase and M phase will just continue throughout the life of that organism. That's it for now. Thank you so much.